Well, thank you all so much for being here. And today I'm going to be talking to you guys about all the work I've been doing over the past several years in establishing systems for studying the evolution of aging and its extremes. And so my talk today starts with my main interest, which is why we age. And now everyone in this audience is acutely aware of the fact that we are all aging at every moment. But this is just not only a fundamental part of the human condition, but it's a fundamental part of life. However, it's also fundamentally a problem. And it's a problem because as we all grow older and older because of the advances in modern medicine, the proportion of the population that is also older is increasing. So take as an example this graph from US Census data showing you the population in the United States broken down based on age categories over time. And what you can appreciate is that the proportion of adults and the proportion of adults over the age of retirement has been steadily increasing both in proportion and in number over time. And this is a problem because over time, then, you are accumulating more and more elderly populations that will be at higher risk of aging related diseases. Cancer is one such aging related disease, and this is data now showing you epidemiological incidence of cancer since 1975 from the National Cancer Institute. And on the x axis, you have the age of diagnosis of every patient and the incidence rate of the population per 100,000 people. And what you can appreciate from this graph is that over time, your likelihood of getting cancer increases and kind of plateaus in old age after retirement. And this is really interesting because not only is this the case for the United States, but this is also the case across borders. And this is also a case across cancer types. So, as I mentioned, aging is a fundamental aspect of all of life. So, not just humans grow old, but also you have baby dogs that grow old and you have baby bats that grow old. We see the aging process unfold across the entire tree of life. And we actually have good data for the maximum lifespan of a variety of species across the globe. So, from this tree of life of mammals specifically, you can appreciate there's a huge diversity of us. But one of the most obvious ones is size, right? But it's not just size. If you plot on the x axis for all the species we have information for, their body size, and on the y axis, their lifespan, you can appreciate this incredible diversity of both traits and the positive correlation between them. Over here, you have your short bodied, short lived rodents, and over here, you have your long lived whales and elephants. So, the diversity of lifespan that we actually see across a tree of life gives us two different approaches for understanding the genetics of aging. The first approach is the traditional one, using short-lived model organisms. And this has been extremely useful for understanding the effects of genetic variants and interventions on lifespan and the mechanisms that govern them. And this is because a short-lived model lives short, so it's tractable to study longitudinally. But the problem is that all the interventions that you're doing in short-lived models are within the context of a short-lived genetic background. That means all of the second order, third order effects of these interventions are going to be in that background, and you're not going to capture the diversity of genes and gene regulatory networks that creates incredible lifespans in some of the longest lived animals on Earth. And so it's always been my core belief that extraordinary problems require these extraordinary solutions. And so in order to comprehensively address the question of why do we age, it's necessary to understand the genetics of extreme longevity in nature to complement the field's work in short-lived model organisms. Now then, a bit about myself and why I'm so interested in aging. I grew up in a really large family in Puerto Rico. And when I say large, I mean my grandfather had 21 brothers and sisters, and my grandmother had 18, just on my mom's side. So growing up, I was unfortunately intimately familiar with aging-related diseases. And it was with this background that then I went to University of Rochester to understand molecular processes involved in aging. This was also critical for my development because I learned about the power of using comparative biology to understand the evolution of complex traits. And it was with that in mind that I went to University of Chicago for my PhD, where I continued to study the comparative biology of aging, now using much more computational methods in it to supplement my molecular background, and studied the role of gene duplication, the exceptional biology of elephants, tortoises, bats, and whales. My approach to studying these extraordinary long-lived groups of animals was very simple. If you think about it, you have genomes for some of these species, so you just comb through the genomes to find longevity-associated genes. You characterize these genes using genomic data sets, and you functionally validate their roles in aging-associated traits such as stress response. Now then, this is a really powerful system, not just for finding cool genes, but for uncovering false negative genes that you would have missed otherwise. For example, in 2018, we published a study about a zombie lift gene in elephants. And this is interesting because we have genomes for elephants and the closest living relatives, manatees and hyraxes. And what we found very early on was that while looking for gene duplications, there's 2 copies of the leukemia inhibitory factor that's shared between manatees and elephants, and they kind of look like a pseudogene. And that's where the story would end had we not had the cell lines for the elephants to do RNA-seq with DNA damaging agents. With this RNA-seq data in hand, we actually were able to show that this wasn't a pseudogene. In the elephants, it's actually a critical portion of their ability to regulate cell death in response to small amounts of DNA damage, which is a fundamental part of their resistance to cancer as a large-bodied mammal. And this story has played out multiple times throughout my career, being able to take these genomes and use these cell lines to actually drive and both inform and drive and validate genomic hypotheses, and not just in mammals, but also across the entire tree of life. But it was also around this time when I became intimately familiar with the 3 horsemen of comparative biology. And so the first of these horsemen is the poor phylogenetic resolution that you sometimes see. And this is a problem because the more separation between your species of interest and its most recent common ancestor, the less resolution you're going to have for detecting aging-associated genetic changes or the changes associated with your favorite traits. Additionally, many of these genomes that we have available nowadays were from the Illumina era, where many of these genomes had suffered from very short contiguity and low coverage, and all these things together creates a whole mess of artifactual genetic variants due to misassembly and reduces the number of genes that were actually captured in the assembly itself, which means you can't even look at those genes when doing comparative work. And all this is bad enough on its own, but if you don't have the third horseman, which is the cell lines, the validated biological organisms that you can use to validate all these things, then it's sometimes difficult or even impossible to confirm or deny any genomic hypotheses, and that leaves your lab in the genetic limbo. So it was with all of this in mind that I packed up my bags and I started my PhD program, my postdoc at the University of California, Berkeley with Peter Sudemont, where I brought with me my passion for aging in bats and whales and set out to establish tools that we needed to actually robustly study aging at all of its extremes. And in 2021, I received the NSF postdoctoral research fellowship in order to establish this system for the functional evolution genomics of longevity in bats. And the reason I chose bats to start is for a number of reasons, not only because they're really cool flying mammals, but there's a lot of bats. Bats are 20% of all mammal species that we know of. And what's one of the more remarkable things is that if you take this positive correlation I showed you guys earlier and do what Austin and Fisher did in 1991, where you regress out the effect of mass of the body size, and you just ask the simple question of how much longer does a species live given what you expect based on its body size, you can appreciate that bats are extraordinarily long-lived as a group, and many species of bats actually live anywhere from 4 to 10 times longer than you would expect just given their body size. Not only is bats interesting for aging researchers, but bats are actually very generally interesting for a wide swath of biologists. And that's because bats have these physiological adaptations to many things such as flight, which includes incredible tolerances to heat and oxidative stress. We can talk ad nauseum about like bats' extraordinary immune systems and their interplay between host viruses and their life histories in these species. In addition, not only do bats echolocate, but what many of you may not know is that many bats actually have social structure, and some species are even capable of kin recognition. And the evolution of these neurological novelties makes them very interesting from a number of different fields. And then there's a whole bunch of other fun facts, such as bats fly while pregnant. And now if you think about it, there are some species of bats that actually triple their body weights when they're flying while pregnant, still lactating, and then also carry their young. So there's a number of cool physiological adaptations in bats that are interesting for a wide swath of biologists. And this system for studying the evolution in bats that I've developed is useful for studying many of these. And in order to establish this system, I actually went out myself into the fields and collected cell lines and tissue biopsies from all these different species of bats. And from these tissue biopsies, you can actually grow all the cells in culture. And the nice thing about doing that is that not only you have a cell line that you can do experiments with, but you have a wellspring of high quality DNA for genome assembly without having to sack the animal, which means you can also combine this with longitudinal studies. So alongside our collaborators, David Inard and Elise Lauterbur at the University of Arizona, we set out to establish these chromosome-level genomes. And what I'm showing you here is a phylogeny of all the bats that have been published to date with genomes, and then some outgroup species. And in color are all the bat species, the 9 bat species that we have actually assembled chromosome-level scale genomes for. And this metric here I'm showing in the bar plot is called the AUNG score. And what this score means is that the lower it is, the more contiguous and complete a genome is. And this black line represents the human telomere to telomere assembly, which is the world's first complete genome assembly. And what you can appreciate here is that not only using this method were we able to create bat genomes that were significantly higher quality than anything published previously, but they are actually approaching telomere to telomere resolution just using the new sequencing technologies that have come online in the recent years. And in addition, alongside our collaborators, we were able to come up with these incredibly high quality genome annotations, which we can use for comparative gene analyses and positive selection scans that we'll be publishing shortly. In addition to all our genomics work, over the past 450 hours and 2 years of field work, I've amassed a collection of over 230 primary cell lines and 33 species of bats. So with this resource, we can actually do rigorously controlled population scale functional genomics in a non-model species that is on par with many of the works that you may have seen in human genetics with quantitative trait loci and the like. So I'm hoping that you get out of this talk the fact that aging is an intrinsically interdisciplinary field. Most, if not all, biological processes you can think of are affected by age at some fundamental level. And there's a very complex feedback loop between internal, external, and evolutionary forces that not only shape a species' lifespan and the genes that govern it, but also interplay between the two. And this fact makes it all the more important to approach aging from all angles in order to untangle this network to understand the independent roles and interactions between genes, environment, and longevity at all levels, from tissues to species. So I've talked a lot about genomic tools. I haven't talked a lot about the tools that the field has developed that will make all this lab innovations possible. And that includes things like next generation CRISPR-Cas technologies to do single cell genetic screens that are massively parallelized. This means that we have parity now between traditional model systems and emerging model systems such as the bats I've developed, where we can do genetic screens to uncover complex gene regulatory networks in our trait of interest, like DNA damage, for example. In addition, for all these cell lines, I've also collected tissue samples from the paired tissue samples from the other wing. And this means that it opens up a whole world of opportunities for us to use functional genomics at scale. And alongside various collaborators such as the Global Bat Network and Bat1K, we're developing these tools to make functional genomics economical at population scales, specifically being able to use existing methylation clocks to get biological ages for all of our bats and integrate all that demographic information that we'll get into our studies at population scales on lifespan. And as more and more chromosome-level assemblies come available, it's imperative that we take all of our existing tool sets and we adapt them to this new pangenome era. The advantage of these pangenome graphs is the fact that you have significantly more power to detect the role of complex structural variations in your trait of interest. And so by taking these pangenome graphs that have been developed and integrating and creating new tools to integrate functional genomic data sets like RNA-seq and these single cell screens, we'll be able to actually validate all of these new structural variants that we're discovering in an unprecedented fashion. And beyond bats, I'm interested in studying evolution of lifespan at all of its extremes. And so I haven't just been working on bats. I've also been working on developing these tools in whales as well and other long-lived animals in order to robustly apply this question across the tree of life. And with all that, I would like to acknowledge all the people who stood beside me and helped make all this possible. This includes the Sudemont lab, my home lab, and especially Peter, John, Stacy, Katya, really everyone in that photo who have been instrumental in me being able to generate all this data and do all this hard work. The Vazquez Medina lab in the University of California, Berkeley is my second home, and there I do most of my work in the cetaceans, which I haven't talked. And I would obviously like to thank my collaborators at the University of Arizona, especially Elise Lauterbur and Joe Suzek, and especially Rose Hadar and Humboldt State for getting me all these resources necessary to build all of these technologies. And I would also like to thank California Department of Fish and Wildlife, CCGP, my various collaborators, and my funding, and above all else, the audience. And if anyone would like to talk more about how you can take this system and apply it to your favorite system and take all these methods and just generally collaborate, here's all my contact information, and I hope you all have a great day.